Reporter constructs are used to measure expression levels of a particular gene, or to determine the subcellular localization of a specific protein. With recombinant DNA techniques, a reporter gene can be fused to the gene of interest, gene X, generating a reporter construct. The genes are fused such that the encoded proteins are in the same reading frame. This ensures production of a chimeric protein containing both protein X and the reporter protein. The gene encoding green fluorescent protein, GFP, is commonly used as a reporter to detect the subcellular localization of a specific protein in live cells. GFP was first isolated from the jellyfish Equoria victoria and provides bioluminescence to this organism. The emission peak for GFP from A. victoria corresponds to the green portion of the visible spectrum. Thus, when cells expressing GFP are irradiated, the fluorescence of the GFP can be monitored by fluorescence microscopy or a fluorimeter. The gene X GFP reporter construct is introduced into cells, and the chimeric gene is transcribed and translated into the fusion protein. GFP protein fusion constructs can help localize a protein of interest. The protein X sequences determine where the chimeric protein is localized, in this case the nuclear envelope. The co-localization of the GFP protein with protein X allows for the detection of the protein within the cell using fluorescence microscopy. When irradiated, the protein emits green fluorescence due to the presence of GFP. The pattern of green fluorescence can be visualized under a fluorescence microscope, thus revealing the localization of protein X at the nuclear envelope. Other derivatives have increased fluorescence, making them easier to detect or emit at a different wavelength and produce a different color, such as the yellow fluorescent protein. GFP constructs have been used to study protein folding and transport and the development of morphological features during embryogenesis, to track specific cell types or diseases, such as cancer cells or viral infections, and to monitor the success of gene therapy. GFP-engineered organisms, such as mice and fish, are being marketed to consumers as pets.